All right, good morning. What I'm going to tell you about is actually a class that we teach here at Yale. So this is a class for undergraduate students, students that are just a little bit older than you are, but they don't have that much more experience than you do. So this is something that is targeted for people who have interest in doing science, who have interest in making observations about the world, but don't necessarily have a lot of experience having done so. And we call it REAL, for Rainforest Expedition and Laboratory. And that's an acronym that I like for a couple different reasons. And the reason I particularly like it is that we want to do something that we think is real science. So there's kind of two experiences that people sometimes have when they get involved in science. And one is that they are given a textbook. And maybe you've been handed one of these textbooks. And there's a bunch of words in it that have bold letters. And what you're supposed to do to become a scientist is memorize those bold letters, right? So you memorize the definitions of all these words. And by memorizing those definitions, you become a scientist. But in fact, if that's what science was, frankly, I certainly would never have gotten involved in it because I really hate to memorize vocabulary. And so instead of that, I'm much more interested in thinking about science as a process. The way we look at the world, the way we study it, the way we understand it, and the way we explore it. And so what I'm going to describe today is the experience that some of these students have had, the process that we've used with some of these students to use or study science as a process, to have them be involved in the actual process of discovery. All right? And so we do this by actually taking students to the rainforest. So I'll talk about that in a second. But a little bit of background about me. We got started on this project because of my dad. So my dad is a scientist. I think the furthest I heard somebody was from here, the farthest away was Maryland. So I'm actually from Montana. So I think maybe that means I win for being the farthest away. So I grew up in Montana. I grew up in a little college town called Bozeman. If you've ever been to Yellowstone Park, it's where you fly in when you go to Yellowstone Park. A little college town of about 25,000 people. And my dad was a plant scientist, or is a plant scientist. And what he does is he studies plant diseases. And at one point, he became interested in the question, what kind of microbes are present within plants? And so he began a process to try to identify and characterize the microbes in plants. And I'll tell you a few of his stories. And so when we were given this challenge of coming up with something interesting to do with students, I thought, oh, how fun would it be to be involved and get involved with my father in doing this project. So that's where that came from. And he's really the inspiration both for this project as well as for my career in science. All right, let's see, what do we have to do here? Okay, so here's what we're looking for. So these are called endophytes. These are all essentially mushrooms. Okay, so if you've gone out wandering around in the woods, or sometimes even wandering around in your yard, you see these mushrooms pop up, right? And they have a stalk, and they have it looks like an umbrella. Most of those are called, the fancy word is basidiomycetes. That's one of these fancy words that you'd have to memorize in a textbook that has bold letters. But it turns out there's another class of mushrooms that are called ascomycetes, and that's what these are. And it turns out most ascomycetes actually don't ever form those, well, they never form those structures that look like umbrellas, that look like mushrooms. They have an entirely different structure, an entirely different shape. Now, these fungi actually inhabit the tissues of plants. Okay? So if you were to take a plant, you would look at it and you'd say, okay, I see this is a tree, this is a stem, this is a leaf. But what we can't see when we look at it under sort of our naked eyes is that actually in between all the cells of the plant are cells of fungi, cells of microorganisms that have become part of the plant. In fact, curiously, if you can imagine, if you were to count up the number of cells that are in you, the number that came from you, and compare it to the number of cells that came from microbes that are associated with you, you are more microbe than you are you. Can you imagine that? Okay? And the same kind of thing is true for plants, that they are chock full of these microorganisms. And so what we're interested in doing is trying to find them and characterize them, culture them, and study them, and figure out what interesting things they might do. So here's a picture of 9 different microbes. These were all grown on the same types of media, same types of Petri dishes. And you can see that one of them makes something that turns the agar completely red. Another turns it completely yellow. They come in all kinds of shapes and colors and beautiful arrangements. And this is what we're trying to find. Okay? Now, one of the reasons we're targeting these is because we think that there's a huge number of them. So these are organisms, they don't cause disease, so we're not worried about something making us sick or making the plant sick. But we anticipate that these endophytes, these organisms that live within the inner tissues of plants, we think that there's about 1 million of them, okay, if you sort of do calculations about what's known and what's still to be discovered. And about, well, the vast majority, maybe 80%, have yet to be found. Okay? So that means that there's a huge amount out there for people like you to discover, people like my students to discover, where we can anticipate that they will go out and they will look and they will find something and it will be entirely new to science, something that no one has ever seen before. And so we want to go to find these, we want to go to places that have the highest diversity of plants, because we think associated with those high plant diversity will be high microbial, high fungal diversity. And so to do that, we go to places along the equator, places that have the highest biodiversity, such as the Amazon rainforest. Okay? So that provides a really easy way for students to begin to do something that involves scientific discovery. All right, so here's how we run the class. We'll do this really quickly. We have a series of goals that they need to accomplish. The first thing they need to do is they need to just figure out, okay, what plants do I want to look for? What plants do I want to look in? And we'll talk about that in a minute. And then they have to go and collect these plants, and then they have to culture the microbes associated with them, figure out what they are, look for something bioactive about those organisms, and then find the chemistry that's responsible for it. Okay? And so we try to do this in the course of about something around 7 or 8 months. So we start in mid-January, and then we finish at the end of July. And by the time we're done, they've done some pretty remarkable things, and I'll tell you a few of their stories. And these are students who are, doing things that, frankly, any of you could do. There's nothing too technically difficult about any of this. All right, so the first assignment is they need to figure out what they're going to collect. What's the project description? Okay? And so they pick plants, or at least write down on paper, plants based on some kind of a theme. All right, so one student said, I want to collect plants that are used as fish poisons. This is kind of a cool story. So what it is if you're, so this is all ethnobotanical kind of ideas. So the native people of the Amazon jungle have lived in these areas for thousands and tens and hundreds of thousands of years. They know what the plants are useful for, and they treat these plants the same way that we would treat our sort of medicine cabinet. Like when you reach into a medicine cabinet, you know what these medicines will do. Well, they treat the forest almost like it was a medicine cabinet. In fact, there's pretty substantial literature out there describing how different plants have different properties and activities. All right, so one of the things they do is there's particular types of plants that if they take their leaves and mash them up and throw them into still water, it causes the fish to become paralyzed, and then they float to the surface, and then they can go fishing with a net and just pull out the fish. Okay? Now, that's pretty cool. That means that there's something in that plant that's causing the fish to have this kind of response, and so that's some pretty interesting biology. So one student targeted those. Another student said, I want to look at plants that are used by the native people to treat snake bites. So if you are in the jungle, the thing that is the sort of biggest threat to you is snake bites, okay? Because there are some really nasty snakes. Now, for reasons I have no idea about, we are now, we've gone on this trip 5 times, and 4 out of those 5 times we have encountered a poisonous snake that is like from me to the people here in the front row. In fact, on the last trip, don't tell anybody who would cancel my funding for this. On the last trip, one of the students actually stepped on a bushmaster, okay? So a bushmaster is the most poisonous snake in South America, and when the sort of local people heard about this, they grabbed a stick that was about as long as this table and tried to get it to leave, but that was as close as they would come to it, and yet this guy had stepped on it unknowingly. So now we call him Bushmaster. So, all right. So some plants, I don't know how effective these plants are being used to treat snake bites, but some plants are used to treat snake bites. We have plants that are parasitic plants. These are like things, one example is something you know about from Christmas. Have any of you ever heard of mistletoe? Okay, so you hang it over your door and then you get to kiss whoever you're standing under it. Okay, mistletoe is a parasitic plant. What it does is it actually jumps onto another plant and grows off of the stem of the plant that it started on. It doesn't form roots and everything else. It's a plant that is a parasite on another plant. One group of students collected plants used to ease childbirth, plants used to treat infections. So these would be things that, used to treat bacterial infections of your skin. One student, and I'm not quite sure how he was going to assay this, but he was going to use plants that were used as aphrodisiacs. So this is something that makes somebody attractive to you. So that, he needed it, I should say. Plants with milky saps, plants used to stop bleeding. And then this isn't always a popular one. Plants used as hallucinogens. Okay? So there's a whole book out there. There's actually a teacher here from Guilford who has a student with the last name Schultes, and her grandfather wrote sort of the book. It's called The Healing Forest, and what he did is he spent a number of years down in South America with the tribal Indians, learning about what plants they use to essentially go into meditating states. So the things they smoke, the things that they take, the things they eat. So there's a whole literature out there on plants used as hallucinogens. All right, so we then have our list, and the students then, off we go. So Yale has a 2-week spring break, so we spend 2 weeks down in South America each year. So in 2007, we went to Peru. So to get to the spot we were interested in, we flew to Lima, and then we flew from Lima to Cusco, and then we flew from Cusco in a little tiny airplane to Puerto Maldonado and landed on what was almost a dirt runway. And then we got out of Puerto Maldonado and went down to the river and then spent 6 hours in the river on a motorized canoe until we reached our destination. So we were so far out of civilization that's the farthest spot I have ever been in my life from anything that looked like civilization. So when we got there, amazingly, there was a little lodge, and we could stay and sleep in that lodge. And so this is the group of students that went in 2007. And then for the past, and that was, as I mentioned, to Peru, for the past 4 years, we've been going to this place, which is on the Napo River in Ecuador. It's not quite as far. We only have to do, 2 flights and 4 hours on the motorized canoe downriver. But anyway, so here's the class from 2008, 2009. And the guy wearing the goofy red hat, that's actually my dad right here. So 2010 and most recently 2011. So we've now had about 75 students that have been going on doing this project with us. All right, now the problem is that none of us are really expert Amazon botanists. So the challenge is that when you get down there and you're trying to find the plant that is the one that's used to treat snake bites, it looks a lot like the plant that might be used for nothing, okay? And so, we don't have to worry. They're not nocturnal. We don't have to go out only at night like we're looking for frogs or something, all right? But the problem is that they're hidden in this sea of greenness. And so how do we find the plants that are the ones we're looking for as opposed to the ones that we don't care about? And so one of the things we do is we actually prepare these flashcards. Okay, so every student is expected to put together pictures of the plant, any kind of information they can gather, and have a card for each one of them. And what's amazing about that is we walk through and talk to people, and they say, here's the plants we're looking for. They can tell you what your collection theme is. They have that much knowledge of what the forest is and what it's useful for, that when you talk to the guides, they can say, oh, you're collecting things that are used for infections. Here's 10 other plants that you should have had on your list. Let's go collect those too. Okay? So we have no idea about what the plants in our neighborhoods do. We've become disconnected from that, right? Whereas they have this very strong connection to what the plants can do, what the roots, what parts of the plants are useful, whether it's the stems or the roots or the leaves. And so it's really amazing to spend some time talking to these people who have knowledge of their environment in a way that we've lost. Now, the other thing we do, so some students are actually able to find their plants using this. But another thing we do is we collaborate with Percy Nunez. And Percy is probably the world's expert Amazon botanist. So he knows his plants cold. Like, you show him half of a leaf, and he can tell you what family of plant it came from. And he knows not only the English name, he knows the name in a variety of other languages. So he's a Quechua Indian, so he knows what it's used for in a variety of different ethnobotanical settings. And he knows the scientific names of many of these. So he's absolutely remarkable. So the students come to depend very heavily upon his knowledge of the forest and what we can do with it. All right, let me tell you one collection story. So I mentioned the Bushmaster. It turns out another snake that is very poisonous, and is another one that we didn't step on but almost did. So we were sort of hopping along the trail, gleeful that we were out in the woods, and one student sort of hopped up and sort of in midair realized that he was jumping over a fertilance, which was this snake. And this one was about 5 to 6 feet long. Okay, this is a big snake. In fact, this is the biggest fertilance that any of the guides had ever seen. So he manages to clear it, and then, of course, then we gather around to take pictures of it. Okay? Now, the lore in the jungle is that wherever you see a poison, the remedy is nearby. Okay, I'm sure it's a hokey lore, but anyway, it's the lore. Okay? So one student was out to collect the tree, the fertilance tree. And the reason it was called the fertilance tree is because the bark of the tree looks like the skin of the snake. So in another example, so we've seen actually 3 fertilances. This is the biggest one. This was actually the second year. The third year we went, we saw another fertilance. It was actually a much smaller one. And in that case, you'll see here's the snake, and not but from me to the front row was the tree. And so here's the tree. It's not quite in focus, but you can see why this would be given the name that it does, that it has the shape, the bark has the characteristic pattern that is the snake. Okay, so one student was able to collect the fertilance tree that they were out to collect. All right, so each student collects something like 25 or 30 different plant samples. We bring them back and go to work. So here's the lab. The students enjoyed this experience so much, they've actually created a huge wall mural that goes up and across the ceiling, side to side in the lab, of the different things they saw while they were in the jungle. So that's kind of fun. There's actually howler monkeys on the other side. And what we're doing now is we're taking the plant material that we've isolated, and we sterilize the outside, cut it up, and then put it onto Petri dishes and ask what grows out. So after we wait a few days or even a couple weeks, and these white fuzzy things start to grow out on the Petri dishes. And those are the endophytes that we're trying to isolate. So at this point, we get the different sizes and shapes and colors that we were looking at. And then when we look at them under the microscope, we see these spores, which is how the fungi reproduce. We see, so these are the different spores. We see hyphae, which are the white sort of stringy things that the fungi have. And so we can begin to characterize them. All right, so here's a question for you. All right, so we have 15 students, 16 students a year that are doing this. They come back with 25 plants. And this is what one student got from what they collected. So in other words, this is all the microbes that one student was able to isolate 1 year. And so you take this and multiply it by about 80. That gives you a sense of how much stuff we've now collected. All right, now some of these organisms no one's ever seen before. Like they're completely new to science. No one's ever described them. No one's ever done anything with them before. And so the question is, what do you do? Right? You can't look in a textbook and say, oh, this is what I should do because this is what the textbook tells me to do. You can't follow some protocol that the lab person says this is what you're absolutely supposed to do. And so the students have to begin to think about, okay, what would be interesting to me? What kind of questions do I want to answer? How could these organisms be able to address problems that the world faces? Like what kinds of problems does the world face that we would like to solve? And how could having greater biological diversity help us solve it? Okay, so let me tell you just a few stories about how the students have applied these organisms to that kind of question. All right, and before I do that, here's the question of what did we find? All right, so don't worry about the details of this graph, but what this essentially describes is what's known. So if you take all the fungi that are known and put them on a graph and compare them to the closest thing that is the closest other thing that's known, you can imagine there's a whole bunch of things that are exactly like other things that have been found before, but there's some weird things, and this is essentially the tail of this curve. All right, so if you found something that by sequence looked like this, it would be, okay, that's an organism that has been seen before. If you found something that was in this range, like the 90% range, this might be a new species. And something out here might be actually a new genus. So the question is, what do the students find? Okay, so if we superimpose that on the first year students, and this is true of the first year and it's true of the most recent year, we find things that have been seen before. We find things that are fairly novel, that this would be a new species. And we actually find things out here that are among the most divergent organisms that have ever been found. These would be at least new genera. We even have some that constitute new order, suggesting that what we're looking at is, as we do this study, we're getting one new genus, not new species, one new genus of organism per student per year. So that means there's a whole lot of stuff out there still to be found. There's a whole lot of stuff for people like you to find if you know how to go look and you know where to go look. All right, so if we have this level, if we have like 15 new genera per year, that's pretty cool. Okay, so like this is a phylogenetic tree. What this means is the length of the line means how closely it is related to the other things on the tree. So for example, 701A is clearly close to other things that are here. These are branches that are pretty short. And everything on this little bud is called the Pestalodiopsis microspora. So clearly 701A is the same thing as what's been seen before. Okay, but what do you do with this? So 1802A, look how long that line is. Look how long the line is before you get to the next one. So you actually have to go down and back up again. And each one of these are different genera. So what do you call 1802A? Do you call it a Masserina? A Pleosporalis? It doesn't actually fit any of those, which means that this student had to figure out what they were going to call it, and they actually had to call it something new. They got to name it. So what do you name something that you've discovered? What do you name it based on? And those are the kinds of questions that the students have to think about. Yeah?
I was about to say, like, your own name.
Turns out that's the one thing you can't name it based on. So the rules for naming new organisms is you can't name them after yourself. You can name them after your teachers, though, which would be really cool. So it turns out a lot of these things end up getting named after Percy. So there's Nunez this and Percy that, right? Because he was so helpful to us while we were in the forest. All right. So having now gone through the process of getting plant material, isolating microbes, figuring out where they are in this tree, and the relationship of how things are related to other things, we now want to do we want to actually look for some things that are useful. So now we'll spend some time talking about screening for bioactivity. All right, so just to keep us remind us what we're doing. We're in the rainforest collecting plants. We bring those home. We isolate fungi. So these are the endophytes. We grow the endophytes up in liquid culture. So we essentially put them into a media. They grow for some number of days or weeks. And they end up in all kinds of colors from gray to black to yellow to red. And then we extract those. So we take the fungi out and we take the essence of what they excreted into the media and reduce the volume down and create these extracts. So sort of the essence of the fungus. And then we screen those for activity. So we have about over 2,000, almost 2,500 extracts now that we've generated over the past 5 years. All right, so what do you hope those extracts can do? That's the question. So I'm going to tell you just a handful of stories of what these extracts can do. And in each case, this is something where the students decide. Like, I didn't tell them, oh, you got to do this, you got to do that. I said, here, you've got access to this, a bunch of new organisms, making new compounds, potentially. What problems would you like to solve? All right, so the first story is Sun Jin Lee. And she was interested in inflammation. She was particularly interested in inflammation associated with preterm birth. So if a pregnant woman gets a bacterial infection, it will sometimes cause preterm labor, so the baby is born too early or too early as a result of that infection. So she was looking for things that could reduce the inflammatory response, okay, the immune response that the body mounts that causes this effect. And so she developed this sort of simple assay in which she put placental tissue into each of these little wells, okay? And then she added the what's called LPS. This is what the bacteria make that cause the inflammation. And then to that she added extract. And then she monitored for the production of a yellow color based on, don't worry about where the yellow color comes from, but that's essentially how yellow it is corresponds to the degree to which there is inflammation. Okay? And here's what she saw. So this is the sort of control. You always want to have controls in your experiments. And then she did the first 10 extracts. And what do you think? Is anything working here? What's happening so far? Is it more yellow or less yellow?
Less yellow.
So we're comparing to this. So who says less? Who says more yellow? Yeah, it's more yellow. So we don't want it more yellow, we want it less yellow. So is anything working for her so far? Total failure so far. Terrible, terrible outcome of this experiment. Okay? She's actually getting things that are causing more inflammation. Okay, so that was the first 10. So then she said, all right, well, I'll do 10 more. So, and then she did this. Okay, anything working here? 1. Exactly, 1, that one. Extract number 8 is working in this case. Okay, so she found one extract from one organism that actually completely eliminates the production of the compounds responsible for inflammation. In fact, when she went through and did the quantitation of this, okay, here's the control, so everything's relative to the control. Almost everything increased the inflammatory response except this one extract which reduced it. All right, well, where'd that organism come from? Well, that organism curiously came from this plant, and it was collected by this student. So this is Corey Eisenman. And Corey went into the rainforest to collect carnivorous plants. She wanted to collect plants that ate insects. That's pretty cool, okay? And it turns out the only carnivorous plant in the area that Percy knew about was the one called Drosera montana, which is a plant about half the size of your palm. So it's a really tiny plant, and it lives on the forest floor in the savanna. Now you might say, the savanna, I thought we were in the rainforest. Well, as you walk along the rainforest on the edge of Peru and Bolivia, which is where we were, there comes a point, like this moment, where you walk out of the forest, and you walk into the sun, and all the big trees are gone, and it turns into this sort of grassy marsh. And then the insects come out. Just nasty, okay? And the forest is actually pretty comfortable when you're in it because it's shady and, it's not, the sun isn't beating down on you. As soon as you walk into the savanna, it's like an oven. It's oppressive, and it's hot, and it's sunny, and then there's insects flying all over your head. And this is where Drosera montana was to be found. And so the goal was to go in and sort of lift up the grass and see if we could find Drosera montana. I gave up after not very long. I'm like, I'm going back into the forest where it's cool. But Corey kept at it, and Percy kept at it, and a few other students kept at it, and they found about 5 of these. They brought it back, and she isolated about 6 different microbes. The fact that it's an E means there was an A, a B, a C, a D, and there's an E, and then there's an F, of which 4 were new species and new genera. Like, no one had ever looked for microbes associated with Drosera montana before, because I'm sure nobody wanted to go through the pain of getting it like we did. All right, so 404E is where that active extract came from. She went on to characterize it. So this is what the spores look like. She characterized the shape of the culture, the shape of the spores. That's what you have to do to name something. And then she named it Aerospheria flavoradians, okay, which in Latin means golden spheres radiating yellow. Now, somebody commented that the thing you do is you name it after yourself, right? And I said, you can't name it after yourself. That's the one rule. Okay, there's a scientific joke here, if anybody noticed it. Okay, let me go back just a couple slides. So the person who was doing this work was named Sun Jin Lee. Anybody get the joke? Scientific joke. They're always really subtle, right? Golden spheres radiating yellow. What do you think of a golden sphere that radiates yellow? The sun. Very good. Okay, so Sun actually did name it after herself, though not quite. Okay, so anyway, so that's now in the literature. She published a description of that. She has credit. She gets to name it. From now on, that's hers. And then she went on to characterize the activity that was responsible for this. So she did chemistry, where you fractionate and purify and analyze. And she ended up getting a crystal structure that shows us that this is what the compound looks like. This is what it looks like if you draw it out. And sure enough, what it does is this is a compound that blocks protein export, so the production of protein and sending it out into the media. So it blocks the production of IL-6 and IL-8. Okay, so that was one story. So this student got a, she got a joint bachelor's, master's degree for this work, and now is doing a joint PhD, MD at National Institute of Health and at Cambridge. All right, next story. So Blair Benham-Pyle, that's her picture up here. Blair heard about this course before we'd even started offering it when she was in high school. And she came to me when she was interested in trying to figure out where she was going to go to college, and she said, I want to come to Yale so I can take this class. I was like, wow, okay. So 2 years later, she showed up. There she was, the first day of class. And I was like, all right, Blair, if you came to Yale to take this class, I guess you'll be in. So, and she did a fabulous job. So she did all kinds of stuff. And she actually just graduated 2 years ago, spent a year doing research, this type of research actually in China, and is now starting a PhD program at Stanford. But anyway, so the work she started on was on hookworm disease. Now, what this is this is a parasite that actually comes up through your feet, if you can imagine. So if you're wandering around in soil that's contaminated with hookworm barefoot, it comes up through your feet, and it goes into your heart and lungs. And the consequence is that when it's all said and done, it ends up in your small intestine and essentially feeds on what you eat. Okay? And a large fraction of the people in Africa and other third world countries are actually infected with hookworm disease that has some pretty debilitating effects on their development. So it means that you end up being iron deficient, and you end up having stunted growth and reduced health. All right, so what Blair was looking for was extracts that could kill hookworm, prevent the hookworm eggs from hatching, or if they do hatch, make it so the larva can't develop. So instead of looking like this nice, I guess if you're a hookworm, this is what nice looks like. Instead of looking like this, if the larva hatch, it looks like something that isn't alive. Okay? And so she screened these extracts, and what she found was one extract that, this is what wild type sort of looks like, one extract that reduced the level of hatching, and everything that did hatch, what the black means, everything that hatched was dead. And she found that if she diluted this a huge amount, it was still active. And in this case, it came from a known organism, at least one that we'd seen before, that was known to the literature. It was a Phaeoacremonium from the water lily, which was in a lake behind the place we were staying. All right, so she went on to identify what it was that was active, so she did a whole bunch of chemistry. So the first thing she did is she got a mass, so this is how heavy, what is the molecular weight? And from that, she could figure out what the ratio of carbon to nitrogen to oxygen to hydrogen was. And then she did a set of things called nuclear magnetic resonance. So she used the instruments in the lab next to where Kurt Zilm works. And from that, she can conclude, actually, that it was, she actually went on to identify what this compound is and show its activity. All right, another story. So in this case, this is Alicia Darnell. And what she found is an organism that you see here. This organism is also novel at the genus level. We haven't named it yet, but this is something that is never been seen or characterized before. She isolated it from a strangler fig. Now, the strangler fig is actually kind of a cool story because it's an example of a pretty nasty plant. So if you're a plant, you have to think about, okay, how do I make it in the world, right? How do I find my place in the world? And how do I get there in the fastest way I can, right? And so what this plant does is it produces figs that birds eat. The birds drop the fig seeds in the canopy when they land. They drop the seeds in the canopy where they germinate, and then the fig sends down a shoot to the ground. But not a root. It's like not a branch, just a long root. And then what it does is it begins to grow on top of the other plant. So instead of making its own trunk, it borrows the trunk of the plant that it landed on. And then as it gets bigger and bigger, it sends down more and more roots to the point, or more and more sort of these long roots to the bottom, to the point where it literally engulfs the trunk of the tree that it started on and kills the tree that it was living on. And so what you see are these strangler fig trunks, okay, where these roots have all fused together, that are just completely full of holes, because it was never a trunk that grew up like the trees we're used to. It was just a bunch of roots that got sent down and then fused together, and so it looks like Swiss cheese. But in the middle of that, it's completely hollow, because what was in the middle of that was the tree that it originally landed on, okay? And so she went out to identify and to find organisms that were growing inside of this tree that was actually killing another tree. All right, so out of this, what she found is that an extract was able to inhibit the growth and actually kill an organism called Candida albicans. Now, this is what causes thrush, causes yeast infections, and if you're an HIV patient, what usually kills you if you die from HIV isn't HIV, you die from thrush. You die from Candida albicans infection. And so we're really interested in finding things that can inhibit the growth of Candida albicans. And so this is actually a really beautiful observation, that we have this really strong level of inhibition at a site where Candida albicans would otherwise be growing. So she went on to do isolation, so fractionating it, purifying it, again doing HPLC, getting a mass spec, doing NMR, and we're fairly sure at this point that we have a new compound. So we have a new organism and a new compound that's active against Candida albicans. All right, so, but not everything is about finding new drugs. These three students were interested in the issues of bioremediation. So the issue is, what questions does the world face? What problems does the world face that we can solve? And they said, well, one of the problems the world faces that we could solve is the amount of garbage that there is out there, the amount of plastic that there is. And what we need are things that can degrade plastic. And so what they did is they set up an assay in which they fed organisms this compound. So polyurethane, which is a type of plastic. And in this case, it's a plastic in the form of a milky white suspension. So they feed this to the organisms, and they're looking for organisms that can degrade the plastic. So what they do is they make petri dishes where the media is, instead of having sugar, instead they feed it this polyurethane. And they look for a place where the organism can grow, and then it has this halo of clearing. You can see where the organism is here, and then there's this kind of clearing zone around it. So that's an example of an organism that's able to actually degrade the plastic. And so they screened all the organisms we isolated for this ability, and they found a handful that were active. They went on to then test them in liquid culture. So here what you see is what we started with. This is the milky white suspension. This is the best thing that we could find in the literature. And then here's the 5 other organisms. And you can see that in some cases, it's completely cleared the suspension. All that's left in here is actually the organism. There's no plastic left at all. So it grew, it made fungus, and it completely degraded the plastic. And furthermore, it could do this where the only carbon that it had, the only source of energy that it had, was the plastic. And it didn't require any oxygen. So it was an anaerobic, sole carbon source growth. So just to sort of emphasize that, here's what it looks like before. 6 days later, here's what it looks like. It completely degraded it. He characterized how the reaction was working using what's called infrared spectroscopy. And I'm not much of an IR spectroscopist. I don't imagine you know much about it either. But if you look at what's on top and compare it to what's on the bottom, I anticipate you can notice that there's something here that isn't there. Everybody see that? And that tells you actually which bond was broken. It tells you that a carbon-oxygen bond was broken during the degradation of the plastic. Now, what John was interested in, he was like, well, okay, polyurethane, that's all fine and good. I really want to degrade polystyrene. Does anybody know what's used to make, what is polystyrene used to make? Yeah. Yeah. Yeah, coffee cups, it's styrofoam. And if you bury a bit of polystyrene in the ground and come back 100 years later, it's pretty much still there. Okay? So he's like, I really want to degrade something that would be difficult to degrade. And so he then figured out a way to screen for organisms that could degrade polystyrene, and he found one. And the data is still a little preliminary, and I don't have a picture of it. But he found things that could actually begin to degrade polystyrene. And so he saw that as a problem that was really important. And maybe he's begun to find a solution for it. All right, so one last story. So, and this actually is part of a story that begins with work that my dad did. And so he actually goes all over the world collecting plant material. I think he's in Australia either last week or next week. Anyway, yeah, he goes everywhere. So this is actually a cinnamon tree in Honduras, if you can imagine. And out of it came this microbe. Now, the curiosity here was this microbe was new. We hadn't seen it before. Again, it's a new genus. But what he'd done is he'd put all the microbes that he had growing and all the plant material he had growing in a plastic box, sort of like this one that's being carried out of the back of the room right now, with a lid on the top. And what he observed is that this organism grew, and everything else that was in the box started to grow and then died. When he went to transfer it and put it on new plates, everything else in the box was dead. So that's kind of weird. So he said, I wonder what the cause of it might be. So any thoughts on how that could work? How could one organism kill everything else that's inside a plastic box? Kind of weird. So there's a smell. There was kind of a strange odor that this thing produced. Okay, and maybe that's a clue. So what he did is a really simple experiment. Turns out you can do really simple experiments. It doesn't all require big, expensive equipment. So this is a simple experiment that you could do in your high school or junior high school class. He takes this new organism and puts it on one side of a petri dish, and then puts another organism on the other side. And what he observes is that if you take the media, so this is the agar, he pulled the agar out in between, this organism is killing this one. This is just a little piece of agar. You can see that nothing's growing out of the agar. This one is killing that one at a distance. It's making gases and killing at a remotely. Okay? So you can imagine that you could actually use this to fumigate without ever actually having one organism come in contact with another or having to isolate the products, right? And so this is something that actually is pretty interesting. So we then ask the question, well, what's it making? And it's like an Aldrich catalog of organic solvents. So there's all kinds of stuff in there. This is the products of what it's making. Now, the next thing we could do is we could actually try to isolate more of these using the first one. Okay? And I'll spare you the details of how we did that. But we used the first one to isolate several more. In fact, we isolate these every year. And so we've now got probably over a dozen of these muscular albus gas-producing fungi that are making volatile organic compounds. And we can show that they're bioactive. So one of the things we want to do is say, well, what's a problem that we could solve? People get lung infections. Particularly, for example, people that have cystic fibrosis get lung infections. And it's really difficult to deliver antibiotics to the lungs. And so we asked the question, could we, in fact, have the gases, because now you're breathing them in, could you actually deliver the gases or have the gases inhibit or kill organisms that are part of cystic fibrosis lung infections? And so one of them we looked at was Serratia marcescens. And so what you see is here's the M. albus on one side of the petri dish, and then there's a physical barrier. And then there's the Serratia marcescens, which is red, so you can easily see it. And you can see here's what it looks like if you don't do anything. It grows quite nicely, and it's red. But if you have one M. albus, you don't see any growth. This M. albus, you see very little growth. Some of them don't produce anything that inhibits Serratia marcescens, and some are sort of halfway in between. So that was kind of cool. And then we said, well, what about other types of lung infections? What about, like, the ultimate lung infection is actually tuberculosis? That's a problem. Could we do something with tuberculosis? So we got in collaboration with a guy at University of Pittsburgh, a guy named Graham Hatfill, who thinks about tuberculosis and works on it. And what we did is we put, so in this case, each one of these set of 6 is one experiment. So we put muscular albus on the outer 4 wells, and then we put media with Mycobacterium tuberculosis in the middle 2. And if it grows, it actually gets kind of thick and hazy. It forms what's called a biofilm. And so you see this grayness. And if you look, here's muscular albus. And look at the middle 2 and tell me, what do you see? Is anything growing here? No. How about here? No. Maybe a little bit here, maybe a little bit there. But the point is that the gases of the muscular albus were able actually to inhibit Mycobacterium tuberculosis in solution, in the media. So that's kind of cool. Okay, so I don't know if that's going to be useful for solving the world's TB problem, but nevertheless, it's an interesting, different way to think about the problem. All right. So in the last 2 minutes, one last story. So this is another organism that we isolated. This one comes out of that same screen where we look for things that can grow in the presence of muscular albus. This one was isolated. It's called Ascocarinysarcoides. And when one of the students in the lab asked the question, what does it make? Okay, so in that we're trying to figure out what the compounds are. What you see is here's what's called the gas chromatography mass spectrometer trace. And each one of these peaks corresponds to a different compound, and then we can go through and identify what those compounds are. Here's the list. And if you do a complete analysis, here's the complete list. So this is an organism that can grow on cellulose and make heptane, octane, 3-octene, 3-nonene, 4-nonene, cyclodexane, decene, cyclodexane, and so on. Do any of those ring any bells? What? Octane. So what is octane? Where do you use octane?
In fuel.
In fuel. So like when you go to the gas tank and you fill your car up with gas, there's like 89, 91, 93. What are those? Those are octane ratings, and the more of the octane there is, the more it costs, right? So what that is that's actually a ratio. It's a ratio of octane to isooctane, or the equivalent of that. Anyway, these are the kinds of compounds, and there's a whole bunch of compounds that are present in gasoline, but these are the kinds of compounds that were present in the gas tank that was in the car that drove you to class today. Now, those compounds and that material was found in the ground and was, pumped and delivered here from Saudi Arabia or up from Alaska or maybe down in Mexico or Colombia, okay? But this organism is able to take cellulose, which is the most abundant form of carbon on Earth, and actually convert it into these products, which could be, in principle, if we can figure out how to do this efficiently, could in principle be used as fuels. And so, of course, that's a pretty interesting thing. So, for example, we then screen these organisms for their ability to produce volatile organic compounds. So Raoul found this organism. Let's see, do I have a picture of it? Yeah, right here. Which makes C9H12 compounds, which is 1 carbon longer than what you get with octane. And so we're in the process of figuring out how we make it, how we can make more of it, and what it could be used for. All right, so the simple idea is sugars in, and then these alkanes out with the application to fuels. All right, so in each of these cases, these are students who are thinking, what's an important problem, and how can I solve it? And how can I use this diversity and these compounds that these organisms diversity produce to find solutions to these really important problems? Because fundamentally, that's what science does, right? There's observation, new discovery, and then you want to apply your discoveries towards challenging problems that would improve everyone's life if you can find a solution to these problems. Okay? And so that's the goal. So like I said, we've walked through the idea of going from a rainforest all the way to the point where we have new chemicals in the test tube that can be used for these problems. So on the last slide, I just want to thank the students and the people that have been doing this with us for the past 5 years. My dad and Percy. This is one group of students. This is another group. One student decided when I wasn't there that he was going to climb a vine. That was a horrifically bad idea. We had a jungle mud fight that was pretty entertaining. So it's been a great, great, great bit of fun. I think some really cool science and some cool observations have come out of that. So I'd be happy to answer any questions. But thanks for coming this Saturday morning.